The next speaker is going to be Dr. Patricia Swart, who is going to give us a talk on IKSA, Decoding South Africa's Biodiversity. Over to you, Dr. Swart.
Is that fine? All right. Hi, everyone. My name is this okay? Sorry. There we go. I'm Patricia Swart, and I am from Diplomix. I am the bioinformatics coordinator, so I coordinate the bioinformatics activities of the network. So for those of you who are not so familiar with Diplomix and what we are all about, we are an infrastructure program supported by the DSTI through their South African Research Infrastructure Roadmap Program, or otherwise known as SARIA. Diplomix stands for Distributed Platform in Omics, and we operate in 3 main domains, those being genomics, proteomics and metabolomics, and then bioinformatics. We are hosted by one of our partner labs, the Center for Proteomic and Genomic Research in Woodstock in Cape Town. Diplomix has 2 main objectives. One is to build a world-class omics infrastructure, and this we do by buying and placing instruments, making sure that those instruments are serviced and maintained, and that they have the technical skills and expertise to be run effectively. Then over time, we've worked on the second aim, which is to enable access to students and researchers to this infrastructure now that we've established it. So this includes expanding our training and outreach, improving our spatial transformation program, so that's bringing omics to areas that are a little bit more isolated, and then our high visibility, high impact projects. This has resulted in impacts such as having available cutting-edge technology in South Africa, having confidence and reliability in this infrastructure, and ultimately higher quality research outputs, as well as skills development and increased awareness about omics and omics technology, improved access and opportunities, and overall world-class reputational gain. So I'm here today to talk about one of our high-impact, high-visibility projects. And in short, these programs we use to develop a community, to increase our training, and to produce methods that are then used or can be used more broadly. So we use them as vehicles to accomplish our broader goals. And today I'd like to speak about the 1KSA project. This is one of our favorites at the moment, and 1KSA stands for 1,000 Genomes of South Africa. Our aim is to sequence, assemble, and make available the genomes of over 1,000 South African species that are important to biodiversity. Why? Having the genome and having this information is like having the blueprint to an organism. You can use that to advance agriculture, to manage conservation strategies, to preserve biodiversity, and enable pharmaceutical innovation. And since South Africa is one of the most biodiverse places on the planet, there are a lot of opportunities in this space. Importantly, South Africa's biodiversity belongs to South Africa and should be researched by South Africans in country. No? There we go. So if you want to go far, go together, right? So on an operational level, 1KSA is made up of these 6 labs, Diplomix headquarters in Woodstock, and the IMBM lab at UWC, Sengen in Worcester, and we have a representative from Sengen lab, Vilku Meyer, in the front row there, and as well as the University of Pretoria Genomics Lab and the South African Institute for Aquatic Biodiversity in Makanda and the SA MRC. And these labs make One KSA a success. So the process starts off with a species nomination, and once that species is reviewed and approved, which includes having all the ethics, sample collection permits, and access and sharing benefit considerations in place, DNA is extracted from that organism, and we can conduct long-read sequencing. I don't want to skim over the DNA extraction and long-read sequencing part because it's a very technical process. DNA long-read, start that again. When you're trying to extract DNA for long-read sequencing, you need intact reads of DNA, and it's quite, hats off to the people in the lab, in short, to get that right. And then we've partnered with Oxford Nanopore Technologies to conduct the sequencing. We have these ProMethine devices in each of these labs, and if you're interested in how Oxford Nanopore sequencing works, please get in touch with me afterwards. Happy to answer questions about that. And they're cool videos and all sorts of things. But once we have the sequencing data from these sequencing devices, firstly, one flow cell can produce up to 2 terabytes of data. It has the potential to generate 2 terabytes. This data needs to be transferred to the compute source. It needs to be analyzed and then stored appropriately. And this is all on the NICUS infrastructure. So I think that's all up to date. I spoke a lot about that when I was here last year, when the conference was at Kruger. And I'm happy to say that in March this year, we officially launched One KSA, and we unveiled our website. And these are some of our latest numbers. We've received over 100 samples. We've sequenced 83, and we've assembled the genomes of 57 species. I encourage you to go look at the website and explore it. There's lots of fun and lots of different factoids hidden around. So please go have a look, oneksa.org.za. And we celebrate the assembly of each of these genomes in the form of a species card, which you can think of as a baseball card for a species. Here are just 4 examples. We have the Cape Gracebook, the red Roman, the Bruder's whale, or the South African whale as some refer to it, and then one of the oxalis species, the many-leaf sorrel. These are on the website, and they're often accompanied by an interview and a short video with the scientist behind the researcher. So yeah, please have a look. It's got some fun stats, and it demonstrates some of the impact. Then in terms of how we are using the CHPC, I'm just going to walk you through some of the steps, very high level. So we get the raw data from the sequencing machines. It gets transferred to the CHPC, and the first step is a bit of quality control where we use nanoplot and nanofilt, and then we also look at, or we estimate coverage and assembly size. That's fairly okay, and we mostly do that on serial long or serial. The next step, the assembling and polishing, is sometimes where we run into a bit of problems. When doing de novo genome assembly, we're not using a reference genome. So it's like building a billion-piece puzzle without actually knowing what the picture looks like. So it's quite memory intensive. And so we use Big Mem for that. And that outputs a FASTA file, so that is the genome. And we then evaluate it using QUAST and BUSCO for completeness and contiguity. And that we can also use serial long for that. So these are just some of our results in terms of the BUSCO scores. So BUSCO stands for Benchmarking Universal Single Copy Orthologs. And so it's a database, and you have your draft genome, and it searches the database looking for expected genes. It will then tell you if those genes are present in a single copy, or if it's duplicated, or if those genes are fragmented or missing. And that all gives an indication of the completeness of the genome. So we've done 35 plants. Of the 57 genomes, 35 are plants. We've done 14 fish, 5 mammals, and 3 little insects. So in terms of some challenges and opportunities, this is, let's start this again. This is a list of the 57 genomes that we've assembled so far. On the x-axis, we have the genome assembly size, which is outputted from our pipeline. And the bars are colored by the expected genome size, which we get off a website called Genome on a Tree. And so to date, the biggest genome that we've assembled is 2.6 gigabases, so 2.6 billion bases. And that's our benchmark. That's where we are at. In 2025, though, this is our to-do list. These species are either on the nomination list, either we've received the DNA, or we've sequenced it and haven't assembled it yet. But you can see that where the benchmark is at 2.6, and there are a few unknown estimates at the top of the graph, but the one that we do know, or we do have an estimate, is the Waterberg cycad, and that has a genome size of 28.8 gigabases, approximately. And then that is followed by, not necessarily closely followed by, but this next big one is the Clivia, and that is 19.2 gigabases. So we want to move that bar from 2.2 all the way to 28.8. That's what we're going to achieve. I'm saying it in a public space, and Vilku and I will make it happen somehow. But I also, some of the challenges that we've experienced are between the computer screen and the chair. So I know that I can shift that line a little bit more. Just spending the last few days chatting with my colleague. So we can immediately shift it a little bit to the right, but hopefully we'll shift it all the way over within the next year. And yeah. So how can you get involved? I have a whole bunch of stickers, if you want. I've dropped them all over the floor on the way up here. There's a whole bunch of stickers. They're free, they're fun, they bring a little bit of joy. And so we all have stickers for our laptops. Then second, you can nominate a species to be sequenced on our website. So if you know someone in the conservation space, or someone who has access to one of the biobanks, or someone who's like super passionate about one particular tiny species that is found on the side of a mountain, let them know about one KSA, let's sequence it, let's preserve that information. And then the next 2 points, maybe a bit of a knowledge exchange. Attend a genome assembly workshop and then in return, help us optimize our pipeline and reach our goal. And importantly, something that I haven't touched on, and maybe should have a lot more in this presentation, but is the open science and then the ethical, legal, and social impact aspects of this sort of project. This data, we're trying to make a digital resource, a digital infrastructure. We want to participate in the global conversation and the global knowledge sharing, but we don't want South African data and South African biodiversity to be exploited by the global north, for instance. So the data need to be protected. They need to be open, but protected as necessary. And then secondly, especially in terms of indigenous knowledge, any benefits that come out of sequencing an indigenous species, those benefits have to filter back into the community where that indigenous knowledge originated. That can be a whole other talk on its own, but it's important to remember. And then one last thing, while I have the stage, I want to mention another high impact, high visibility project that we have going on, and maybe I can share some of this at next year's conference as well. But this is Ingwe. Ingwe is also using Oxford Nanopore long read sequencing. However, it's in the human genome space, which comes with its own set of challenges. And the aim of Ingwe is to speed up the diagnosis, the time to diagnosis in the rare disease space. So literally changing lives and saving lives. It's a bold statement, but what was said yesterday, having that vision and then the courage to do that, with the support from NICUS, that gives us a little bit of confidence and courage to take on this project. And all of this infrastructure will also be, all of this work will also be made possible by the NICUS infrastructure. And thank you very much.